Hello, welcome to Physiology on Demand, where today we are going to learn about neuronal structures and location in great detail. Start with a little bit of review and then get in some new content so we can dive deeper into our neuronal physiology. And so this picture is just kind of a precursor for our next slide, where I'm going to do just a little bit of a review about pumps and channels. And I know this is information you learned for test one when we talk about the plasma membrane, but in this case, I'm going to apply it directly to the neuron. So please don't skip ahead, but listen for its specific application. So pumps, as you recall, require energy and are going to move things against a concentration gradient or up the gradient. And in the neurons, we're going to have our typical sodium-potassium pump, but we also have calcium pumps. We haven't talked much about calcium pumps, but calcium pumps are really important not only in the neuron, but also in muscle to reduce the level of calcium in the cytosol, because it's really important to keep the cytosol levels of calcium low so that when we have an influx of calcium through a calcium channel, we then will have the reaction either in the muscle or the neuron to have the release of a neurotransmitter or to have the start of excitation coupling cross bridge to occur. That's all we need to know about pumps. As far as channels, they do not require ATP. They are facilitating diffusion, allowing substances to move down a concentration gradient. And in the neuron, we are going to have leaky channels. We're going to have chemically gated channels, which you may have learned as ligand channels. Those two terms are synonymous with each other. Chemically gated, you can call ligand. Ligand, you can call chemically gated. No deduction for using either term, and I will use both terms during this course. And then the third type are going to be voltage gated channels. And then there's a fourth type I will mention briefly at the end. Leak channels are passive channels that are always open, and they allow a continuous diffusion. And examples of these are going to be sodium channels, which are going to allow sodium, which is extracellular, to slowly leak into the cell, and potassium, which is mostly intracellular, to slowly leak out of the cell. And that's why we have to have the sodium-potassium pump constantly moving those substances in the opposite direction. As far as ligand channels, these are normally closed. And in response to the binding of a neurotransmitter, they will temporarily open, allowing their specific ions to diffuse. So we can have a ligand-gated sodium channel, we can have a ligand-gated potassium channel, and we can have a ligand-gated chloride channel. All three of those exist in the neuron. For voltage-gated channels, these also are normally closed, and once again, they will temporarily open, this time in response to electrical charges, allowing their specific ions to diffuse. And examples of these are sodium-gated when we have depolarization, the sodium-gated channels open. When we have repolarization, the potassium-gated channels open. And the calcium channels open, and those are really important as far as allowing the influx of sodium, which is going to allow the synaptic vesicles containing the neurotransmitters to release the neurotransmitters. Okay. And then in some of our sensory neurons, on their dendritic endings, we have modality-gated channels. Okay. And they function in response to very unique sensory stimuli. For instance, in our eye, the presence or absence of light has them function. We have temperature changes that can have them function. We have pressure, the absence or pressure of continuous pressure or deep pressure. We have pH changes that can do this. So these are very specific ones. And when we go through our or special senses, I will talk about, for instance, with vision, how these work. Okay. So don't worry about those unless I specifically talk about those later on. We will also be talking about how CO2 and pH work with specific central and peripheral chemoreceptors and baroreceptors to keep your body in homeostasis. But that's much later in the course. All right. Let's back up and look at these voltage gated channels, because there's one type of voltage gauge channel that is different from the others, and that is the sodium channel. And even if you took anatomy with me, I didn't tell you that the sodium channel is special, okay? Whereas the other ones are either open or closed, the sodium channel actually has two gates in it, and each gate can be open or closed. So you can have both gates open, both gates closed, or you can have one gate open and one gate closed. And because of that, the sodium channel can be in three different states. And so I'm going to show the picture and then I'm going to tell you how this works. So here we've got three different states. So in this first one, we can see extracellularly, we have sodium on the outside. Here at the bottom, this is inside the cell, and here's the plasma membrane in the middle. And so when you look at the sodium, we have what's called an activation gate, and then we have what's called an inactivation gate. And in the normal state, what happens is the activation gate is closed and the inactivation gate is open. And because the activation gate is closed, sodium cannot enter the cell. And that's why most of the sodium is outside the cell. Now, when the cell reaches threshold, there is rapid opening of the activation gate. Now, remember, the inactivation gate is already open. And now the activation gate opens in response to the threshold. And so since both gates are open, sodium can enter the cell. So that's the second state that the voltage gated cell can be in. Now, the third state is the activation gate is still open, but the inactivation gate, temporarily closes for milliseconds. So this rapid opening at threshold, this rapid opening of the activation state also triggers this inactivation gate to slowly make its way up here and block it. But then the weight kind of pulls it back out. So it's only closed for milliseconds and it can't be stimulated to reopen. So how does this look with the activation energies? At resting potential minus 70 millivolts, the activation gate is closed. As it's depolarizing from minus 50 to positive plus 30, both gates are open. And from peak plus 30 going down to minus 70, the inactivation gate is closed. And then this one will open and the activation gate will close and it will return back to the first state. So most of the time sodium can't enter, but it's for two different reasons, two different gates. All right, so I hope you understand that. All right, so now let's look at all these different channels and pumps and see where they are in the neuron. So let's start first on this left side where all this yellow part is of the neuron, okay? Because some of these pumps and channels are all over the place and some of them are only in specific areas and it's all related to the function. So in this yellow area, what we can see throughout the entire plasma membrane of the entire neuron, we are going to be having our sodium leak channels and our potassium leak channels the entire length. So sodium is going to diffuse in, potassium is going to diffuse out. And in addition, we are going to be having the sodium potassium pump, returning the stuff where it belongs. And all of this is going to establish and maintain that resting membrane potential, which I will be talking about in more detail in the next video. Okay. Then if we look up here where the dendrite and the cell body is, this is called the receptive segment of the neuron. And up here we have chemically gated channels. Specifically, we have potassium channels, chloride channels, and what we call cation channels. And what a cation channel is, it's one channel that lets sodium go in and potassium go out in the same channel. Okay. So those are the three types of channels that are found up there. And this also lets it establish and maintain the resting membrane potential. Continuing on in the neuron, the most important part of the neuron for action potential is this area right here, the axon hillock, which physiologically is known as the initial segment because this is where the action potential is generated. So if you are asked, where is the action potential generated in the neuron? It is the axon hillock. That's where the action starts. Okay, that's where the nerve impulse is started. Doesn't start in the dendrites, doesn't start in the cell body, it starts in the axon hillock. And this has both voltage-gated sodium channels and voltage-gated potassium channels. And those are the exact same type that are going to continue the entire length of the axon and also all the way down through the axon terminals. And those are where the action potential continues to be transmitted. And so those parts of the neuron are known as the conductive segment because that's where the action potential is conducted. All right. So that continues all the way down to these little knobby things at the end of the axon terminals, and they have a specific name. And their specific name is the terminal bouton, also known as synaptic bulb. So pick which one. Personally, I like going from the axon hillock to axon terminal, so the terminal boutons, which just means terminal buttons. It's just a French word, just sounds really fancy, terminal bouton, but you can call it synaptic bulbs if you prefer. And here we have a change. Here we have voltage-gated calcium channels, and you should remember this because these are required in order to get the calcium to enter the synaptic knob, which is required when we have that massive influx of calcium. That is the signal for the synaptic vesicles filled with that neurotransmitter acetylcholine to undergo exocytosis and release those neurotransmitters into the synaptic cleft where they're looking for their receptors on the postsynaptic cell. Okay. If you don't have that influx of calcium, then acetylcholine cannot be released. But that then gives us the problem that we have all this calcium in the cytosol there in the synaptic knob. And so that is why we need a calcium pump, which is going to use a bunch of ATP to send that calcium back outside the neuron. It needs to be kicked outside the neuron so that we can have another action potential come down and bring it in so you can release more acetylcholine. All right, so hopefully now you understand all that. So let's look at a different picture of the neuron and relate all this to voltage and current and all that kind of stuff I mentioned before. So if you recall, voltage was the difference in electrical charges. And when we talked about the neuron, we said the outside of the cell had a very positive charge, whereas the inside of the charge was extremely negative. Yeah, it wasn't having a good day. It was very negative. Okay. And when we looked at this, this gave us resistance, which was the opposition to the movement of the charged particles. And basically, all this resistance was due to the plasma membrane there. All right. And so, in order to have this action potential. We had to generate a current. And what the current was a movement of charged particles across the plasma membrane through these open channels, through these sodium channels and potassium channels. These were voltage gated sodium channels and potassium channels. And that gave us a current. And so I think you understood all this. And so now we're going to throw in that little physics thing, Ohm's law. Okay. Now, I'm not going to ask you what is Ohm's law, but I know that we've got a bunch of people in physiology who love to throw in Ohm's law when they're teaching the lab. So, in case your lab professor is one of those people that asks you, what's Ohm's law? Now, I've told you what Ohm's law is, which basically it tells you that current technically is voltage divided by resistance. So, it's the difference in electrical charge divided by the resistance. Okay. So what does that exactly mean when we're talking about a neuron? So let's look at this neuron. We had this neuron, and in this neuron, according to the previous slide, we talked about we had all these leaky channels of sodium, all these leaky channels of potassium, and we had all these sodium potassium pumps. And based on all of that, we talked about we had the neuron had a resting membrane potential of -70. So basically, the current maintained the neuron's resting membrane potential of -70 through all these leaky channels and pumps. So that is where we were. That's the bottom line. All right, so take all this information, the previous slides, and look at this overview. Read all these words. Make sure you understand it. Make sure you can apply all these concepts and build on what you learned previously in anatomy. When I was reading a lot of the answers from the concept from the quiz questions number 1, I noticed a lot of the answers you were just giving me the answers that you would have learned in anatomy and not diving deeper. This is not anatomy 2.0. This is physiology. You need a deeper dive in physiology. So I'm challenging you because I know you can do it. All right, have a great day, and I will see you for the next video. Thank you for all your hard work, and I'll see you soon. Bye.